Everybody to this Google Hangout, our first Hangout for Foundational Neuroscience for Perception and Action. I know if you joined our Hangout live, but for those of you who may be watching and thinking about joining, let me just say just a few things as we get started. This is really time for you to ask questions that will help you advance yourself in our course, but perhaps you also would just like to ask a question of a more nature. That's perfectly fine. But what we would ask, because we do have quite a number of people that were interested in the Hangout, is if you ask a question when we call on you, free to follow that up with a second question if you wish. But once we have addressed your questions, we would ask that you just step out of the Hangout so that someone else can join. I'll just remind you that there are slots for 8 of you to join the Hangout at one time. Well, everyone can courtesy to everybody, see if we can't get as many people through as possible. Those will be the rules that we'll abide by. Thank you very much. And Dee is with us, and maybe I'll turn to Dee first. Your microphone is, so please unmute yourself and go ahead and ask the question this Dee, I think you need to scroll towards the top of your window where you should find an unmute button.
Hello?
Yes, hi, thank you. I can hear you.
This is Sairam. I'm doing it. Okay. Sairam, yeah. This is, good evening, sir. I have a couple of questions for you. The first question is, that is the human accelerated regions, and what role do they have in our human body? Second question what I have is, sir, the major causes of numerous neurodegenerative diseases and dementias, such as the Alzheimer's, is accumulation of compounds, the peroxynitrates, which in turn affects neuroglial cells of that nature. So can you suggest me some compounds which may either inhibit the activity or may act as scavengers? Actually, I have compiled a scientific note of these things, so I would just need your tips so that I can arrange things.
Questions. Let me address the second one first. I don't have very many suggestions that come to my mind immediately. I'd have to look at this just a little bit, but I would direct you to a post in the discussion forum that discusses a paper from that provides some insight, I think, to the question you're asking. There is a study on an animal model to suggest that there are local inflammatory factors that might be more important than had been previously. Arginine, the amino acid arginine, might be promising as a novel intervention that might delay local inflammatory factors. So my guess is that there will be a number of agents that are capable of modifying this local inflammation that might become very important as a therapeutic avenue for looking at degeneration, not just of the Alzheimer's type, but perhaps other forms of neurodegeneration that might convert the same basic mechanisms. So that would be one tip. And I'm sorry, Sarah, I didn't quite catch your first question. Could you put that to me one more time, please?
So my first question was about HARS. I remember reading an article, I think it was by Catherine Pollard from, I think, California, Columbia University to be precise, where she described about the specific regions in the brain which I think are protein R1 deals with dexterity of our fingers. We can move it like this, or it helps in writing and all that. So my question was, what is the role or the impact of these regions on our normal actions? Another request is, can I mail you the document of that dementia thing if it's possible? I mean, this directions.
Well, I'm sorry that I don't think I have much to say about your question, Sairam. I'm sorry about that, but I bet there are others, of course, who could provide you with more information. So my suggestion would be to put this question out on the discussion forum and let's see what happens. That will give me a chance to dig into some of my resources and give you a better response than what I feel I'm capable of doing at the moment. So please go ahead and do that, and hopefully we can have a good set of discussion points to share with you. So thank you. Where are you, Saram?
So I'm calling from Delhi.
Delhi.
New Delhi.
Well, thank you very much for an hour in the morning, as I imagine it must be where you are.
Thank you. Good day.
Great. Thank you. Gabriella, you've joined us. I think you were the first or second person in. You're currently muted, so you can go ahead and unmute yourself and go ahead and ask your question. Gabriella is writing something to us, which should be fine. Okay, I have a problem with the microphone. Okay, well, hopefully you can get that sorted out. And meanwhile, maybe we'll go to Modesta.
Gabriella, if you look on the left side, you'll see a button that says Chat on the very top. It's a little blue icon. If you click that, you should be able to go ahead and type your question.
Yes, for other folks that are out there, there is a group chat feature in the Hangout, and even if you're not joined with us live, you still can go ahead and type a question using the so that would be true for those of you inside the Hangout as well. So how about if we go to Modesta? So welcome.
So welcome, but actually I don't have any particular question like now, so I just join.
Okay, where are you?
I'm from Poland and I'm not a medical student, so I'm just interested in this course, so I'm just visiting and see how it's looking.
Good. Well, I hope you're finding some use for the course, and I hope that you're finding some of the lessons interesting and the discussions that are ongoing to be both fun and engaging, but also informative. So thank you.
Yes, it is actually.
We've been really pleased with the responses to the Let's Discuss forum.
Okay.
I think there's been some really interesting back and forth, and it's been, I think, a really wonderfully rich dialogue. So we look forward to having further discussions in the future. Well, feel free to hang in here with us. We have room for other students to join us, so we don't need you to step out necessarily. But if you do have a question, the chat function, or just indicate some other way, and we'll try to get back to you. But maybe at this point we can move on to Gustavo. Are you there, Gustavo? Gustavo has joined the chat, or joined the Hangout, but microphone is muted. Okay, so we've just seen Gabriella's question. So Gabriella says, I wish to know a little more about the connection of olfactory, second cranial nerve, and brain beyond smelling. Sorry for my English, no problem at all. That's the first cranial nerve. Okay, the second cranial nerve is the optic nerve, a sense of sight, but the first cranial nerve. So we're really not focused on the olfactory sense in this course, and only because I wanted to keep the course as short as I possibly could. So that's an omission. So I'm very grateful for the question, Gabriella. We can talk more about the sense of olfaction. Olfaction is one of the oldest senses, phylogenetically speaking, evolutionarily speaking. It is a privileged sense in a very interesting way. The first cranial nerve, the olfactory nerve, connects directly to the telencephalon, that is, to the forebrain. So the connection goes to the olfactory bulb, which I think you saw in one of my videos on the ventral surface of the brain. The olfactory bulb looks like a nerve, but it's not a nerve at all. It's really the brain. It's an extension of the brain. So the olfactory bulb, in turn, connects to parts of the cerebral cortex that are somewhat primitive, but highly conserved across the mammalian and even the vertebrate nervous system. These parts of the brain are intimately connected with behavioral responses involving the hypothalamus, involving parts of the prefrontal cortex and the brainstem. And our sense of smell can be a profound motivator of behavior. This is especially true in animals that use chemical signals as a way to mediate relations. Now, there's a lot of controversy about whether we as humans do that as well. Some would say yes, others not so sure. But it is the case that our olfactory sense has direct access to parts of the medial temporal lobe, such as the amygdala and the hippocampus, that are involved in emotion and memory, respectively. So the sense of olfaction, I think, is very interesting for many, many reasons. But it is somewhat privileged among the senses in its direct access to circuits in the brain that can have a very profound influence over cognition. So that's just a few thoughts that come to mind about the olfactory system, and if you have more particular thoughts, we can get in there and talk more directly. I see you have a comment about memory. As you may know, it's quite common that a sense of smell can be well integrated into human memory. Sometimes we have particular memories of smells from even our childhood, many, many years ago. And this reflects the direct access that the connections of the olfactory bulb have with our medial temporal lobe memory system. In fact, one of the main sources of input to the hippocampus that mediates memory is derived from the parahippocampal gyrus, in a division of the cortex there called the entorhinal cortex. Well, the entorhinal cortex gets direct input from the olfactory bulb. So the hippocampus is only a few synapses removed from the sensory neurons that first interact with odorants in the airstream. So there is an extremely tight relationship between the odorant signals that are first processed by the brain and their potential to have an impact on memory, both as an influence over the formation of memory, but also as a trigger motivating their recollection. Recall memories were re-engaging the sensory circuits in which those memories were first established. And the intimate connection between the medial temporal lobe memory system and the olfactory cortex is part of the explanation, I believe, as to why memories and olfaction seem to be so tightly linked. Certainly the cosmetic industry understands this very well. The sales of perfumes and related substances that can have a powerful effect on our consolidation of new memory, but also of past memory, has had a major economic impact on that particular industry, at least. So thank you very much, Gabriella. I hope you get your microphone situation sorted out. You can join us a little later. I'm looking to see if Gustavo is present, but evidently not. We just see a big red screen on our end and a muted microphone. So if Gustavo is there, perhaps he can use the and post a question, or at least let us know what's going on.
Well, we got a couple of questions from the Q&A from David R. Baczynski. His question is, how does maternal mom and parental dad DNA differ in the contribution to functions of various regions of the brain? Catherine Dulac has some mice humans.
David, thank you for the question. This is a really active area of investigation. We do know that there are some genes that are linked to the maternal line that can have a profound influence over behavior. This seems to be particularly true in the hypothalamus. I don't really know a whole lot about this area, but I have learned a few things from some colleagues here at Duke about this. So it is, we think, one of the means by which the epigenetic influences of parenting in early life can have a profound effect throughout the life. And those factors that are derived from the maternal line seem to be particularly modifiable in this regard. So again, I'm not an expert in this field, but I do know enough to have a sense that this is an active investigation. And there seem to be certain genes that are derived from one parent or the next in terms of what's actually functionally expressed. And the hypothalamus seems to be a fascinating part of the brain with respect to a preferential expression of certain types of genes that can have broad influences over behavior. And what I find particularly fascinating is some of these genetic factors can be modified through epigenetic means and can have long-lasting and even lifelong-lasting impacts. Certainly in the animal models in which these have been studied, like mice and rats, we can see influences that can be traced not just to the mother, but even the grandmother. So that's interesting to consider, the influence of interacting with one's grandmother, and see how that might even play itself out over the expression of genes in such an important brain structure such as the hypothalamus. So I think I'll leave it at that. Again, I don't know a whole lot about this field, but thank you for the question. Maybe Nick can point us to something else.
Okay, this one just came in, and I actually just read this yesterday. So this is from Rakesh. I want to know Professor White's height hit mainly from the perspective of rescue motor functions.
Head transplants. You know, I saw the thread, but I have not really gotten into it, so I don't know the specific details of the question there. Is this more of just a thought experiment about head transplant? Can you shed any more light on that, Nick?
So apparently there's I believe he's an Italian doctor, and there was a volunteer, I believe he was from Russia, and I can't remember which disease he has, but it's like just basically destroying his body, and he's volunteered to basically transplant his head on a good donor body. Somebody had posted up in the forum, and I watched a TED talk on it. He said he should be able to do it within like 2 years. Yeah,
right. I'm just double checking the post now on the discussion forum. Yeah, this is still in the realm of science fiction more so than the realm of actual neuroscience. You know, I can anticipate that there could be some preservation of function that relates to just doing the appropriate vascular surgery, but in terms of actually establishing neuronal connections, a real tough challenge to try to resolve, in large measure because the white matter itself is expressing signals that are suppressing the growth of axons across a lesion of some sort. So really the challenge is, how do you connect the caudal medulla to the rostral spinal cord in such a procedure? So I'm fairly confident that the vascular surgeons can do the job of the non-neuronal aspects of the surgery. The question would be, how can one possibly connect the long tracts of the brain stem to the spinal cord, even if one could deal with the obvious immunological issues that are attending any kind of organ transplant. So, you know, that's one level of concern. Another level of concern is much more ethical, even philosophical, and that is what would it mean for cognition to be embodied in someone else's body? One of the fascinating domains of study, I think, is this notion of what does it mean for our brain to have a body and interact with that body, to communicate with it, and to be influenced by it. And in real sense, our whole sense of self is a creation of the brain. And to put that cognitive organ now into another form seems to me to be really problematic at many, many levels. So even if the immunological, the cell biological, and the molecular challenges could be overcome, I think we would still have to tackle the ethical, moral, and philosophical challenges that such a hypothetical procedure would impose. You know, we know from such surgeries, as radical as they are, involving hand transplants and face transplants, that there's some very real cognitive consequences of these kinds of procedures, and I can hardly even imagine what we might be facing should the science progress where we can overcome the restrictions that the molecular and the cellular factors impose currently. So I think I should stop rambling about that issue, but it is one of those shocking new suggestions that remind us that we really live in incredible times with respect to biomedical technology. So I think Gabriella was chiming in on this as well via the board. What do you think about the head transplant? I was wondering if they could fix the connection between the brain and the spinal cord. I think we're making incremental progress on that, particularly in the preclinical studies with animal models, but still a long ways to go in order to overcome these inhibitory signals that are expressed by particularly white and oligodendrocytes in particular. You know, one of the thoughts that we have about all of this is that the nervous system goes through such incredible effort to organize itself to perform function that there's a cost to that. And I think part of the cost is consolidating growth and development and not allowing that degree of order to be easily disrupted. And if it should be disrupted, one of the costs is that it's very difficult to reestablish that order and connectivity, given the mechanisms that are in place to secure the result of brain development in the first place. So that's, I think, one of the challenges we face when we're dealing with a nervous system that is as old as we are. So maybe that relates to a prior question that I think it was Samar had about various factors that contribute to neurodegeneration. We have to remember that the brain cells that are with us today are, by and large, the ones that were born in the second trimester of our own gestation. And so, you know, those neurons are vulnerable as they age as a result of the various influences of the chemistry of the body, as well as the own metabolic byproducts that they produce in life. Well, Gustavo has chatted in to say that he is present. He is a teacher. Thank you very much for contributing to the education of young people. Obviously something I'm very much grateful for and appreciating of for anyone who wishes to do that. But Gustavo's having some problems with the microphone, and he's just here listening to us. So thanks for joining. If any of you out there want to go ahead and join us, but meanwhile, maybe I'll ask Nick if there are more questions we can cover.
Yes, this one's, oops, this is from Jenna. Hi everyone, congratulations for the amazing course. My question is, what happens to the brain immediately after?
Okay, thank you, Joanne. Thanks for participating in Foundational Neuroscience and for joining us here in the Hangout with your question. So right after a lesion, there are a number of things that happen. So if the brain is experiencing, let's say, a focal injury, let's say a trauma, there is a dramatic change in neural activity, that lesion, whatever it may be. So one can to be immediate impacts on the firing patterns of those neurons that have been traumatized. Now, with that trauma, there most likely is also going to be a disruption in blood flow. So within seconds to minutes, hours, that's impactful. So the lack of oxygen, glucose that might be part of the injury, whichever injury process you imagine, be it trauma or stroke, that will have a huge impact on neuronal activity and the integrity of neurons. And as their integrity becomes compromised, the activity increases. So one might see a focal seizure, one might see hypersynchronous patterns of fire. As these things unfold, there can be some real dire consequences for neurons that are in the region of focal ischemia. Even beyond the region of direct insult, these activity patterns can also lead to the expression of genes that can actually lead to the destruction of the neurons. And this unfolds over a matter of hours to days. Cells can continue to die over many days following even a focal injury. So there's both bad news, obviously, but also good news, because this protracted period of cell death provides opportunity for therapy and for interventions of different kinds. We have a therapeutic window with which we might develop new strategies for trying to preserve these neurons that might otherwise be on a path of their own destruction. Now, for those neurons that survive this initial acute phase of injury, we can expect there to be some recovery and even some synaptic plasticity, some circuit growth, some reorganization. And the more we study this, I think the more impressed we are with the capacity of the brain to reorganize itself following a focal injury. And this is certainly more robust in young brains compared to older brains, but I think it's pretty clear now that throughout the lifespan, there is the potential for circuits to become reorganized following a focal injury of some sort. So there's really a cascade of events that happen, beginning with alterations that can ultimately lead to the reorganization of circuits surrounding a region of what is probably going to be a region of necrosis at the core of the infarction or the core of the trauma, whatever it may be. So there's much we need to learn about this. We need to understand how to modify this plasticity for therapeutic benefit, how we can shape that in a more constructive, productive way, because not all modifications are necessarily adaptive. So we want to make sure that as individuals recover from their brain injuries, they are receiving the right kind of rehabilitation at the right dose and structured in the best way to really optimize functional recovery. So that's a question we address a bit more in depth in medical neuroscience, but in foundational neuroscience, we're not going to try to get too clinical in our considerations, but I appreciate the question nevertheless.
We got another one that came in. It's from Zohabib, because he had a problem with some confusion on the quiz. Asked about a cranial nerve that causes the condition known as strabismus. So that confused him. Since there are two cranial nerves that deal with the eye, how do you know? Oops, where'd it go? Which one deals with the condition like this?
Yeah. So the condition that the question is referring to is strabismus. So strabismus refers to a condition where the two eyes are misaligned. That is, the two eyes are not converging at the same point. Bonding in the two eyes would be co-activated by the same stimulus. And the consequence of this could be double vision, or with prolonged strabismus, a consequence could be amblyopia, which means a loss of vision in one eye. And this is not a problem in the eye, it's a problem in the brain. So how does this happen? How might the eyes be misaligned? Well, they can be misaligned if there is a biomechanical problem with one of the ocular muscles. As you may recall, there are 6 pairs of extraocular muscles that rotate the globe in those 6 muscles are innervated by 3 cranial nerves. I believe the question suggested 2. There's actually 3 cranial nerves. Cranial nerves 4 and 6. Now, if I'm recalling the quiz question correctly, you're going to be looking for whether cranial nerves 3 or 6 are provided as options. And damage to any one of those three nerves could be sufficient to produce strabismus. So sometimes, as a clinician, we can do a simple test of one's eye movements. We can simply ask a person to track a finger on the horizontal plane, in the vertical plane, and then in any oblique axis, and try to look at the rotations of the globes. And if you know the muscles and their innervation patterns, you can deduce whether there is an injury to one of those 3 pairs of cranial nerves. So as you review that quiz, perhaps you'll want to give it another go. Look for a response option that relates to one of those 3 cranial nerves that supplies innervation to extraocular muscle, 3, 4, and 6. Okay, are there other questions there, Nick?
There's quite. This one's showed up a lot in the discussion boards. Apparently, there's a lot of non-medical students out there. So this one's specifically from Doc. He would like to know the best way for non-medical students to learn this course. Much of good research comes from interfaces between sciences. So basically, it's how can non-medical students succeed in this course?
Right. So this course really does make some assumptions about your background, obviously. It is helpful to have a general background in biology and anatomy and physiology, even human anatomy. And I realize that that's not the case for many of you who just have an interest in the brain, but don't really have the background that someone who might be interested in medicine or medical school might have. So I think you're doing exactly what I would suggest. You're participating in the course, you are engaging in the discussion forum, and I think I would simply encourage you to continue to use the discussion forum as the principal means by which you can support your learning and seek connections from the content of foundational neuroscience to your own background, whatever that may be, and your own area of interest. Now, when there are particular questions that arise or particular issues that support an additional background, I would encourage you to use the course wiki as your primary resource. You'll find in the course wiki a long set of study tips that should be very beneficial. And I think someone who does not have a background in biomedical science can probably benefit from those study tips. So please go ahead and check out that resource and see if that can be helpful to you. But also just feel free to draw upon the strength of our global learning community, because you're not alone. There are many out there that share your concerns and your interests. And so I hope this can be a course for those folks that aren't particularly interested in clinical applications of foundational neuroscience, as well as those who are. So thank you again for your question and your post.
This one's from Isabel Cardenas. She's a psychology major student. She's interested in neuroscience. Could you please, she's got 3 quick questions. Could you please explain top-down and bottom-up processes? How do glial cells work, and how does Alzheimer's disease change the brain?
Okay, let me first take the top-down, bottom-up. I've to avoid that language because I realize it's jargon and it can be difficult to understand. But essentially, what we refer to as top-down means commands or influences that are derived from the cerebral cortex and that are concerned with cognition, executive functioning, planning. So these are influences that are derived from the more of the executive centers of the brain that can be communicated to a lower level of processing. Sometimes that lower level is also anatomically inferior, that is, a structure that's more closely related to processing that's going on in the brainstem or the spinal cord. So sometimes when we say top-down, we quite literally mean top-down, both functionally and anatomically. Sometimes when we say top-down, we mean this in more metaphorical terms, as in the influence of the executive control networks over, let's say, lower-level sensory processing. And this is where attentional mechanisms might have some role to play, top-down modulation of lower-order function. Now, bottom-up is complementary in the sense of building up from a more primitive level of processing or more basic level of processing to a higher level. And that's easy to understand in the context of something like the somatic sensory system, where we can think of stimuli that might begin in the contact surfaces of the skin, arising and being transduced into electrical signals that then are processed through the nervous system in a bottom-up fashion, meaning from the periphery towards higher central levels of processing. So let me move on to the second question. Would you rephrase that again, please, Nick? I didn't quite catch it. Something about glia?
Yeah, glial cells.
And what specifically?
How do they work?
How do glial cells work? Well, there are essentially three major classes of glia and maybe additional subcategories that we don't need to mention, but there's a type of glia that makes myelin in the nervous system. In the brain, that is the oligodendrocyte. In the peripheral nerves of the body, that's a cell called the Schwann cell. And this cell really has that as its primary function, to make the insulation around axons that can facilitate the propagation of electrical signals over the length of an axon. There are other molecular mechanisms associated with these oligodendrocytes, some of which I alluded to earlier, that have to do with regulating the growth of axons. But for the mature nervous system, I would say that their primary function is to insulate axons through the formation of myelin. Another type of glial cell is the astrocyte. This is a small star-shaped cell, which is why it's called an astrocyte. And this is present primarily in the gray matter of the central nervous system. The astrocyte has numerous functions. It serves an important role in the uptake of neurotransmitters and neurotransmitter precursor molecules that are present in the synaptic cleft. It also has a role to play in balancing the ions and some other small molecules that are present in the extracellular fluids around the synaptic compartments. Astrocytes have a very important role to play in formation of the blood-brain barrier. And in recent years, that they have an important signal with surrounding neurons and synapses. That communication function might actually modulate the way neurons are conveying their signals. And in the context of plasticity, the astrocytes have an important role, we know, to shape the formation of new synaptic connections. So that's a very exciting discovery that is up on. And the third major class of glial cell is the microglial cell, which has conventionally been thought of as an immune cell to inhabit the central nervous system, primarily as a macrophage, but it also seems to have other functions that allow it to retain a kind of a memory for immune activity throughout the lifespan, or at least throughout the life of the cell. And this is very interesting. We know that, for example, early insults nervous system through disease or through infection or toxin can have a long-lasting impact on the macrophages across the lifespan of the organism. So that's, again, a very new and emerging area of research focused on the interactions between the developing brain and the immune system. And the third question, Nick, what was the third of?
How does Alzheimer's affect the brain?
Yeah, how does Alzheimer's affect the brain? Another huge question. Alzheimer's disease is a neurodegenerative condition that causes neurons to die, particularly in parts of the brain associated with what we call the limbic forebrain, but also the association cortices of the temporal, frontal, and parietal lobes. It's a degenerative disorder that seems most severely parts of the brain that are involved in making memory. Loss of short-term memory is one of the cardinal signs of Alzheimer's disease, and as the disease progresses, that allow a clinician to achieve some confidence in the clinical diagnosis of Alzheimer's disease. Although canonically we would say that the diagnosis must be confirmed at pathology to be certain of the diagnosis, and what the neuropathologist would look for is the presence of microscopic features in the brain tissue itself. And they take the form of extracellular deposits of amyloid protein and a variety of other proteins and compounds that are associated with these local regions of inflammation that we call plaques. In addition to these plaques that are rich in the beta amyloid protein, there are changes in the cytoskeleton of neurons. And what is observed is a tangle of neurofilaments within these neurons. And for that reason, the second hallmark of Alzheimer's disease is called the neurofibrillary tangle. So the presence of both beta amyloid plaques and neurofibrillary tangles would confirm a diagnosis of Alzheimer's disease upon autopsy. And then the final feature I would mention is that with this ongoing neurodegeneration, we often see a loss of brain structure. So there can be significant atrophy of certain regions of the brain. This question is bringing to mind a brain that I removed with some students in one of our cadaveric anatomy courses last year, and the degree of atrophy of the cerebral cortex was almost unbelievable. I have never seen a brain so degenerative in all of my years of teaching and all of my years of interacting with human brain specimens. So it just reminds me of really what a devastating condition can be, and how much of the brain can actually degenerate in life. And of course, my expectation would have a fairly limited cognitive, but I did not know the person in life. Their cadaver came to us through the Duke Anatomical Gifts Program, their generosity and the generosity of their families. But the upshot is that there could be widespread degeneration in those parts of the brain that are involved with cognition and memory in particular. So thanks for that. Let's see if we can move on to another.
All right, so Camille's sent a couple, and he sent one to sort of explain a little better. Trying to find it now. Says, even as until now we have seen only a small part of intricate anatomy and functions of the brains, it is obvious that a tremendous order and immensely complicated process is acquired during evolution. Basically what he means is, has it been considered the approximate time of natural evolution for the acquisition of an immense order observed in the brain functioning? Is it a compulsory evolution of brain complexity, or is it a causal case? It keeps flipping on me.
Yeah, I think I get the gist of the question. There were these epochs of fairly dramatic evolution of brain and body, and something happened around the time of the extinction of the dinosaurs, about 65 to maybe 85 million years ago, where there was this massive radiation and a huge divergence in the size and complexity of mammalian brains. It's not so clear yet from the genetic and the fossil records exactly what were the selective pressures at play there, but we can imagine that there were some fairly dramatic changes on the planet that might have set the stage for such incredible divergence. So prior to that time, at least going back yet a few 100 million years, the mammalian body and the mammalian brain seemed to be relatively stable. So mammals were small creatures on the order of probably just a few tens of grams, and even less than that for the most part, with some exceptions, but mostly small-bodied creatures with relatively simple, relatively primitive brains with very limited cerebral cortex. And then again, about 85 million to 65 million years ago, there was this dramatic increase in the complexity, particularly of the cerebral cortex. And that explosion really began at that period. And with respect to human evolution, we know that there were ongoing changes throughout the last 500 to 1 million years of primate evolution that eventually gave rise to the hominids. And even among the hominids, there has been significant change, both an expansion and in some cases even a contraction of brain and body. So this is a fascinating story. It's a topic that I did not devote time to in foundational neuroscience, or even medical neuroscience for that matter, but perhaps that's something that we can get into in future editions of the course. So I appreciate the question. It's always fun to think and talk about the evolution of the brain. We have to be mindful of the limits of our understanding of our own natural history, but with additional developments, particularly in the area of genetics and molecular science, I think we're gaining much greater insight into our own history as a species and our kinship with other mammals, not so different from our own. So thanks for that question. Nick, another one?
Yeah, Gustavo actually just sent one in, maybe tie into what you were just talking about. It says, what happens to your brain or mirror neurons specifically if you grew up in a white room without stimulation or with a pretty different species from yours?
Thanks, Gustavo. This is an interesting question. I actually had a discussion just this morning with a colleague about what seems to be a bit of a conundrum, and that is that if you consider maybe you've gone to see animated movies, even with very fancy computer graphics, and it seems like our computer scientists and graphic artists still perfected the representation of the human form in virtual reality. So despite how beautiful many of these products are, there's still something that we notice isn't quite right, is sometimes disturbing. Now that's sort of one observation. Another observation is how readily we can detect biological motion, even from the most primitive cues. Perhaps you've seen examples of this where people have been filmed with maybe just a few points of light on their body. So movement patterns, for example, could be reduced to the motions of just a few points of light. And yet we can so readily interpret that as meaningful biological motion. So here's the disconnect. On the one hand, we are great at interpreting biological motion, and on the other hand, when we're presented with what we might think of as more information in a rich illustrator or graphical artist's view of a human form, we're very sensitive to the dissonance and to the imperfections. So what's going on here? Well, I don't know. I don't think anybody really knows, but what I can imagine is happening is that our mirror motor system is tuned to biological motion that we have observed throughout our lifetime. And if it were possible to do the experiment that you're suggesting, as terrible as it is, to grow up in a white room or in an environment without that instruction, then my guess is that we would be impaired in our ability to understand the actions of other people. And there is some suggestion that perhaps something akin to this dysfunction in the mirror motor system underlie some individuals with autism spectrum disorder that seem to be deficient in their ability to interpret biological motion. That motion that's derived from socially significant movement, including motions in the region of the eyes and the face. Our mirror motor system is likely to be present at birth, but certainly is educated through our interactions with other human beings and other agents, not just humans, but non-humans as well. And that the experiment that you're suggesting would likely lead to a mirror for the minimal representation of such motion, and likely have consequences for social and neurocognitive development. Nick, do we have time for one more question?
There was one quick question from Isabel. She was wondering if she could share the YouTube link for her psychology class.
Oh, absolutely. Isabel, feel free to make use of any links that you find associated with this course, and even any assets that I've made available to you, including the videos themselves. So that's part of my motivation, is to help people learn, and I'm happy for you to be a medium for conveying the impact of foundational neuroscience. I would just ask, of course, that you respect whatever copyright issues attend what you have in mind, but in terms of just providing links, that is certainly fully within our fair use doctrine, so I'll do that. Well, I think we are just about out of time here, unless anyone wants to join us at the last moment or post a last question to the chat board in the forum.
There are quite a few.
Okay.
I can't get to.
All right. Maybe I'll just ask Nick to pick out one more question and we'll conclude on that.
Okay. Let me look at it real quick. Hold on.
Well, while Nick is looking, Gabriella has written on the chat board a question about someone who loses vision at different ages. I assume that you're referring to what I was speaking about concerning the influence of vision on the development of the mirror motor system. I think there is likely to be a for the tuning of the mirror motor system, and with loss of vision after that critical period, then there's likely to be adequate. I would suggest still some ongoing plasticity, maybe even some impairment that might set in later in life. But I think one is likely to be more successful following a lesion that might have had its impact following the critical period than during that critical period. Now, someone who is simply born with a dysfunctional visual organ, let's say due to a cataract or something of that nature, then it may be that the mirror motor system could be tuned by other kinds of sensory inputs, including sense, the vestibular sense, and the somatic sensory or proprioceptive senses, perhaps even the sense of olfaction, something you were asking about earlier. So critical periods are an important aspect of brain development that we're not going to discuss in foundational neuroscience. So if you're interested in that topic, check out medical neuroscience, where we take on the issue of brain development across the lifespan.
We'll just do the short, quick one. Could you, this is from Philippe, could you just give a short survey on your own research? What do you consider as the most important contributions toward a better understanding of the brain?
That I've made myself, or generally other
Yes,
folks?
you yourself.
Well, I think, well, thank you, Philippe, for that question. Difficult question to answer, but I'll try it. Maybe what I'll say is the contribution that I'm most proud of, and it actually has to do with what I was just referring to, the concept of critical periods in early life. I guess about maybe 8 or 9 years ago, my colleagues and I came across a discovery that I think has turned out to be quite significant, and that is the influence of early visual experience on the development of the circuits in the brain that are very important for orientation selectivity and direction selectivity. And in particular, what my colleagues and I have worked on in the last 5 or 6 years is the importance of real motion at a very early stage in postnatal life. Now, we're working with an animal model in our simulations that have helped us to understand the mechanism there, but I think the upshot is that for a world with some deficiency in visual function, there is a very critical period, and it might be as short as just a few weeks to months, where the brain is going to require motion stimulation. And so if a child is more important to try to provide clear vision for that child at a very early age so that these motion discriminating networks can develop and such individuals can have the benefit of motion perception throughout life. Research in an animal model might suggest a permanent deficit in motion discrimination without the benefit of this early exposure to motion patterns. So that would be one response to this question. So thank you very much. Well, I think we need to conclude there. Thank you for your interest and your questions, and we would like to do at least one more Google Hangout in the remaining weeks of Foundational Neuroscience. So I invite you all back, and if you are just checking us out online, feel free to indicate your interest to us through our next survey about wanting to join us. So thank you, Nick, for helping me here in this room at Duke, and thank you all for joining us online. Look forward to interacting with you in the discussion forums and in our Twitter account. And otherwise, I wish you a very productive and successful final few weeks of medical neuroscience, of foundation for perception and action. I knew I was going to get the two courses mixed up at some point. Pardon me for that. Okay, so I'll see you all next time. Thank you.